this was a bit of a CTE study, and they were looking at stages of CTE and how, because what happens in CTE is that you have, your brain has infoldings, right, called sulcuses or sulci, and then you have the crests around in the brain. And CTE tends to form neurofibrillary tangles or collections of tau in the sulcus, kind of at the low point in the depths. They call it in the depths of the sulcus. And it doesn't tend to form it at the crest. And so this study was trying to figure out why and what are the differences between the tissue in the crest and the tissue in the sulcus. And what are the differences in patients that have no history of head trauma and no neuropathological diagnosis versus those with repetitive head injury, which was this group, where they have a history of playing football, but they don't have any CTE. And then they have the CTE group where they have a history of playing football and they also have a diagnosis of early stage CTE. So they were looking at what genes are located in these two different areas and what other type of inflammatory and biological or metabolic markers are in these two areas that differentiate why CTE may target the sulcus versus the crest. So here's the controls here. So they didn't play any sport. All of the other groups played sports. So this is repetitive head injury. This is CTE. So you can see the numbers here. 6 in the control, 5 in repetitive head injury, and 10 in the CTE group. They had years of exposure to football right here. And then they had their RIN, which is the RNA integrity number, because they were looking at RNA. It basically means what was the quality of the RNA they were able to gather. And basically this just shows that the quality was similar through all the groups. And that's kind of the point of that right there. Here's what they did. 50 micrograms of fresh frozen tissue taken from the dorsolateral frontal cortex gray matter at 2 locations for each case. They looked at the depth of the superior frontal cortical sulcus, and they looked at the neighboring gyral crest at the level of the anterior caudate nucleus. mRNA was extracted and isolated and converted to DNA using a reverse transcriptase and then sequenced to look for the differences in the genetic material, the genes between those two separate tissues. And here's what they found. They found a complex inflammatory response and gene expression differences in the two tissues in controls, in the repetitive head injury group and the CTE groups, but it was kind of on a continuum, whereas the controls had less differences, RHI had more, and then CTE had even more. But even in the absence of repetitive head injury or CTE, the sulcus had a unique expression pattern versus the crest. This may be why different areas respond differently. There may be different cell types in these areas or different concentrations of different cells, which means that they may be more susceptible to the shearing forces, maybe more susceptible to inflammation, et cetera, and that may be why we see the patterning we do. When examining how the sulcal-specific alterations might change in response to trauma and disease in the repetitive head injury group and the CTE group, there was a complex mixed increase and decrease of inflammatory processes such as astrocyte development, scavenger receptor activity, regulation of immune responses, leukocyte migration, antigen processing compared to the controls. So here's the control group. They had altered genes where the different genes were related to development and homeostasis. In the repetitive head injury group, the altered genes were related to metabolic and mitochondrial differences. And then in the CTE group, they were related to immune function and inflammation. They were mostly there repairing tissue in the CTE group, whereas in the RHI group, they were still in the metabolic phase. So there may be some continuum, whereas the control group has, differences in genetic processes, genetic tissues there, genes that are in those tissues. When you have some head injuries, those genes are now responding in different ways to basically the metabolic end of things. And then when you progress into the CTE, now it's more inflammatory and immune type of situation. So this just kind of points to maybe the pathology of CTE has something to do with the way that we respond to the injury, both in a metabolic and inflammatory process, right? So getting back to those NFL players, those recruits that don't have really good, strong molecular makeups, maybe we can do something to counteract that and make sure people are healthier coming in.